welcome everyone. Thanks for joining us today for the Tools and Technology Seminar. For those who are in the room, if you have questions at any time, of course, just raise your hand, ask questions as you normally would. For anyone who's online, if you have questions, you can put them into the chat box. I'll be monitoring that and I can let our speaker know if any questions come in. I don't think I have any other announcements, so I can just introduce our speaker. Today's speaker is Anthony Cicalo, who is a grad student in DCMB in Indica Radiopaxi Lab.
Thank you. Hi, my name is Anthony Cicalo. I'm from the Radiopaxi Lab. I'll be giving a presentation on the bioinformatics of multi-way interactions in the genome. So for my outline of this presentation, I'll be giving a brief introduction to genome architecture. I'll also talk about two different technologies, one being Hi-C, and this captures pairwise interactions. The other being Por-C, which is an extension of Hi-C. This captures pairwise interactions as well as multi-way interactions. I'll also talk about my ongoing work as well as my future work. And my goal is that by the end of this presentation that you guys have an understanding of what we're doing with the Por-C data that we're generating in the Radiopaxi Lab. So the human genome is made up of 3 billion base pairs on 23 chromosomes, on 23 chromosome pairs that contain over 20,000 genes. That's 3 billion letters, and if you were to take these out of the cell, that would be approximately 2 meters long. So everybody knows, everybody's familiar with what the textbook structure of the chromosome is at mitosis, but what's still unknown is how the chromosome is organized during interphase. And that is what these technologies are trying to deliver, is characterizing what is going on in the chromosome during interphase. So one technology that does this is Hi-C, genome-wide chromosome conformation capture. Hi-C provides us with pairwise contact information via proximity ligation of chromatin that are close in space. And this is what a typical Hi-C contact matrix looks like. It's very diagonally dominant, and these contacts are close to the diagonal are considered intrachromosome interactions. And what intrachromosome interactions are contacts within one chromosome or the same chromosome. Contacts far away from the diagonal are interchromosomal interactions. And these are contacts between chromosomes of two different chromosomes. And this is a limitation of Hi-C, is that you don't really pick up much of these off-diagonal contacts. So graph Hi-C versus hypergraph Por-C. Hi-C provides us with pairwise relationships, and this is actually represented as a graph, and its matrix is an adjacency matrix. Por-C is a multi-dimensional relationships, and this is naturally represented as a hypergraph, and its matrix is an incidence matrix. A hypergraph is a generalization of graph in which a hyperedge can join any number of nodes. And a hypergraph can represent multi-dimensional relationships unambiguously. The unique thing about a hypergraph is that you can do what's called click expansion and decompose these multi-way interactions into their pairwise contacts. But you cannot go back from the pairwise interactions back to its multi-way interactions. So that is why we like to use Por-C. And I will be using this paper, Deciphering Multi-Way Interactions in the Human Genome, which we've just submitted to Nature Communications, and it's in the bioRxiv right now, to explain the way that we are visualizing these multi-way interactions from Por-C data. And I just want to note that this paper was featured on Oxford Nanopore's webpage last week. So this is a figure from the paper. In panel A, this is the workflow of Por-C, and it's showing how Por-C can capture pairwise interactions just like traditional Hi-C, as well as these multi-way interactions, like a 3-way concatemer and a 4-way concatemer. In panel B, this is a visualization of a multi-way concatemer. And these are the incidence matrix. In the left-hand column is the genomic loci, or where that concatemer comes from. And then these are all of its various binning at high resolution to low resolution. And this is real data from an experiment on adult fibroblast cells from chromosome 8. Here in panel C, we are showing a visualization of a hypergraph and its corresponding incidence matrix. You can plot these multi-way interactions and their incidence matrix. For instance, this orange 4-way interaction is this square right here. And we can see that it has 4 contacts, 3 of which come from the same chromosome, which are intrachromosomal interactions, and one contact is from a different chromosome, which is the interchromosomal interaction. And as I said before, we can decompose these multi-way interactions into their pairwise interactions and then plot them as a graph or an incidence matrix. And then it's interchangeable with its high C contact matrix once you decompose these multi-way interactions into their pairwise contacts. And this is what we have in our lab. We can start from cell culture and sequence it all by ourselves in the lab using this machine. And this is what allows us to generate the long reads and have the multi-way interactions. So this is the workflow of the 4C. These first 4 steps, cross-linking, restriction, enzyme digestion, proximity ligation, and reverse cross-linking, these are all typical of any high C or 4C experiment. Where it differs from a typical high C experiment is right here. With 4C, you're size selecting for long fragments. You want fragments that are greater than or equal to at least 1,500 base pairs. And you're also using nanopore specific adapter sequences, which allow that sequence to attach to the nanopore and transverse through the nanopore that it can be read. Another thing to note, different than Illumina, is this is single strand sequencing. This is not double strand sequencing like Illumina, but you do get single strand and you get very long single strands of DNA. And then we data process it through a pipeline, and we return 2 different types of files, one being a contact table, and this gives us all the pairwise interactions, and the other being the align table, which naturally comes to us as an incidence matrix. Another thing to note from 4C versus Illumina is that we can generate these 3-way concatenamers that can be decomposed into their pairwise contacts. With a typical second generation sequencer, you get A and B and C as the pairwise interactions. But actually, when we get this 3-way concatenamer from a 4C read, we can decompose that into AB, BC, and the additional AC read, which you would not see in the second generation sequencing. So why are multi-way interactions important? They allow us to identify what we call transcription clusters. This is similar to the idea of transcriptional hubs or transcription factors. Some definitions to keep in mind is a transcription cluster is a group of genomic loci that co-localize for efficient gene transcription. Master regulator is a self-regulating transcription factor that binds to its gene analog. A specialized transcription factor is a transcription cluster where at least one master regulator binds. So what we do is we get these 4C reads, and then we query 5 kb to the 5' end and 5 kb to the 3' end, and we look to see if there's any chromatin accessibility from a tag seed. And if there is, if all those genomic loci are accessible, then we look and see, well, does this have any POL2 binding? And if at least one of those loci that is accessible has POL2 binding, we come down here and we put that into a list that we call a potential transcription cluster. Once we have a subset of potential transcription clusters from the 4C contacts, we then look and see, are there any genes expressed? And we're looking for there to be at least 2 genes expressed in that multi-way concatenamer. And if there's at least 2 genes expressed, and then those 2 genes have a common transcription factor among each other, we consider that a transcription cluster. Once we have transcription clusters, we look to see if that common transcription factor among each other is a master regulator, which means it can bind to itself. And if it is, then we call that a specialized transcription cluster. And that's why multi-way interactions are important. We can identify these things from data. And this is the algorithm I walked you through. I feel like it's much simpler to walk through the algorithm using this chart than using this algorithm. So these are some transcription factors that we identified from data. Okay, neonatal fibroblast data set, an adult fibroblast data set, and a B lymphocyte data set. And each of these transcription clusters up here has a gene that is unique to this data set. not every gene in the transcription cluster is unique, but at least one of those genes are unique. And so we can see here the edges correspond to chromosomes and that have a gene associated with them. And then the red is the transcription factors that combine to at least 2 of these genes. And it's not a requirement that every edge has a gene. Like right here, for instance, this is a 5-way concatemer that has 2 genes associated with it and has these common transcription factors, but it doesn't have a gene on every single edge. Okay, so I'm now going to transition to single cell 4C. Just, keep in mind that everything that I talked about before comes from population 4C, and it comes from human cell lines. Everything I'm going to talk about now is single cell, and it comes all from mouse cell lines. So everything with single cell is similar for the first 4 steps, but what's different from single cell high C is we don't use PCR amplification to get enough genomic DNA to sequence. We use what's called multiple displacement amplification. And this allows us to have long reads, and it also allows us to not have so much amplification bias that PCR would give you. Multiple displacement has been proven to not amplify the same strain of DNA over and over again, where PCR has been shown to do that. So that's one big differentiation from single cell high C to single cell 4C. Also, we received 24 cells from Florida State. We're doing this single cell project in collaboration with Dr. Peter Fraser from Florida State. And so he sent us 24 cells using 3 different amplification kits. 8 cells were amplified by QIAGEN, 8 cells were amplified by Cytiva, and 8 cells were amplified by NAB. So what we did was we sequenced all these cells and plotted them, and we could see right away that NAB was just very sparse. It was diagonally dominant. There wasn't very many off-diagonal contacts, and it was just sparse in general. But then Cytiva and QIAGEN, you know, this cell right here and this cell right here look pretty similar, but uniformly there's some sparsity that we didn't see with Cytiva. So we ended up choosing Cytiva as the MDA kit to go with. And some other statistics that we did were we calculated the number of total contacts that come from this kit. And then we also filtered it to only be unique pairwise contacts. And we noticed that Cytiva also provided us with more unique pairwise contacts after filtration. And also, we saw a very significant number of off-diagonal contacts, these interchromosomal contacts, which we like to see with this technology. And also, we saw 3.56% coverage of the mouse genome with Cytiva. We saw a relatively high coverage with QIAGEN too, but again, the uniformity with Cytiva just showed us that this was a more promising kit than QIAGEN was. And we loaded all 8 of these cells on one flow cell. And the N50 of the read length was around 5,000 base pairs. So we had a pretty long read length as well using these kits. I just want to touch on that. At 4C, you can represent them as hypergraphs. They naturally come as an incidence matrix, but we also can decompose these back into their pairwise contacts and plot them as a high C matrix. So that's what we did. And this is what single cell 4C looked like, not using the filters. So that's what we decided. We need to build a tool to actually visualize this data and make some analysis from it. So we built this single cell 4C filter. And what that consists of is we're only allowing cells that have a quality score of greater than or equal to 9. We're only allowing the frag counts to be less than or equal to 4. And we're only allowing the map quality score to be greater than or equal to 30. So why we chose those parameters is the quality score is the confidence level the sequencer has that it correctly called the base. The frag counts is the number of reads containing a fragment. And why we made this 4 was these cells are not synchronized. We don't know what stage of the cell cycle they're in. So it's possible that they could be in mitosis and have 4 copies. So we're allowing that. And then map quality is used to quantify the probability that a read is misaligned. So what we use is BWA-SW. We've also tested many other liners, one being Minimap2, using the CW5 parameter. We chose this parameter, which is a very small K-mer window, with advice from Dr. Hing Lee, who developed both of these aligners. And he decided that that one might be the best for looking at single cell data. We've also created a stringent filter, and this is just only looking at the unique paired contacts. So it's you reduce the amount of contacts there are from just this filter. And then we built a binary filter, so it's an unweighted filter where it's 1 if a contact exists in that band, 0 if a contact does not exist in that band. And just a remark is if the pixel is black, there's a contact that exists between those two bands. So this is what single cell 4C looks like filtered at 1 MB. And this is what it looks like unfiltered. So unfiltered, it seems as if all chromosomes are interacting with each other. Once you've used a single cell filter to visualize it, you don't see that. Also, Dr. Peter Fraser has been working with this cell line for a while. He actually developed single cell high C using this cell line. So we just wanted to compare single cell 4C to single cell high C, and that's what we did. And then this is a more representative example of single cell 4C versus single cell high C. Single cell high C being the lower triangle, single cell 4C being the upper triangle. This is chromosome 1, this is chromosome 11, and this is bend at 1 MB. And this is what I want to point out is the fact that we are getting more off-diagonal contacts from single cell 4C that you just don't see with single cell high C. So this is another representation of single cell 4C, chromosome 1, bend at 1 MB. This is 40 bend window down the diagonal. This is a 196 by 196 matrix. And what this is showing is one read from 4C and its multi-way interactions in red. So this is for all 40 bends. One interesting thing is that we can pull out that concatemer. So this just happens to be a 6-way concatemer, and this contains the gene ST18 from the mouse genome. Just want to remind the audience that high C naturally provides pairwise relationships, which is an adjacency matrix, and 4C naturally provides information as an incidence matrix, and that we took that incidence matrix and decomposed it from the hypergraph into the pairwise contacts to plot these. So we like to use the incidence matrix. We just did that to compare this to the single cell high C to see if we could get any resemblance of what a single cell high C experiment looked like using single cell 4C data. And this is intrachromosomal incidence matrix from chromosome 1 bend at 1 MB. This is the incidence matrix where the columns are the concatemer, the rows are the loci, and this is showing bends 4 to 195. This is a 196 by 196 matrix. And this is showing the contact order bend at 1 MB. There are a lot of two-way contacts, but just keep in mind that this is bend, and we still are seeing a significant number of multi-way interactions in single cell 4C. And this is just the same thing for chromosome 11, where the columns are the concatemer and the rows are the loci. And then this is between chromosome 1 and 11. So I have my ongoing work is I will use single cell 4C data to identify transcription clusters very similar to the way that we did this in population data. Right now I'm sequencing neonatal fibroblasts using single cell 4C. So this will be a very nice comparison to the previous paper to look at population neonatal fibroblasts and single cell neonatal fibroblasts. And then I'm also working with Dr. Wishaw, who's the pioneer of cancer stem cells. with the VARI 068 cell line to sequence these as single cell 4C data. And this is done by Dr. Reed. This is Sky, and this is a normal cell line, and then this is the same cell line cancer. And what we see is this chromosome aberration, chromosome 1 aberration. And we're very interested to see if single cell 4C will show us this at a higher resolution. And thank you to everybody that's helped me in the lab. Everybody has contributed in this project in some way or another. And I'm thankful to be working with these people. And if you guys have any questions, that's the end of my presentation.
If anyone has questions, either in the room, you can raise your hand, or online, you can put it in the chat box, or maybe use the Zoom reactions to raise your hand and then you can unmute yourself.